Good evening. Thank you for attending our Ahead of Cancer lecture series this evening, sponsored by the Allegheny Health Network Cancer Institute. We are excited to welcome Dr. Nathan Bahary with us this evening to talk about clinical trials. There are a few housekeeping items that we'd like to review before we start. During the presentation, all participants are able to type questions into the chat portion of the Zoom connection. We will be addressing those questions at the end of the lecture if we have time. Participants are not able to speak during the conference, but again, we encourage feedback via the chat. Welcome, Dr. Bahary. Nathan Bahary, MD, PhD, is the Division Chief of Medical Oncology at the AHN Cancer Institute and Director of the AHN Cancer Institute Clinical Research. Board certified in clinical oncology, he received his medical degree from Cornell University Medical College in New York, now Weill Cornell Medical College, and his PhD from the Rockefeller University in New York. He completed his residency at Beth Israel Deaconess Medical Center in Boston and a research postdoctoral fellowship at Harvard Medical School in Boston. He is a member of the ECOG GI Committee and the NCI Pancreatic Task Force. Dr. Bahary's research interests include identifying novel genes and pathways underlying normal gastrointestinal development in cancer to both design rational molecularly based disease markers for early detection of gastrointestinal cancers, as well as rational directed therapies for their treatment. Tonight, we'd like to welcome Dr. Bahary.
Well, thank you very much, Crystal. I very much appreciate it. And thank you all for taking time out of your evening to listen to us talk. And I hope you'll make it interactive by putting questions, and we'll try to answer them at the end. What I really would like to do in the next half hour or so is really give you an overview of a bit at the beginning of the history of clinical trials and where we're headed with these and how we're trying to use clinical trials in order to develop new therapies for cancers. So first of all, I'm a consultant to a number of pharmaceutical companies which bring in trials here. I will not discuss off-label drug usage at all. And so this is the goal of what we're trying to do, not just the goal of tonight, but it's to understand how we can advance cancer care. And clinical trials are really the means that we use to identify the newest, the next generation of treatments, new things for cure, new things for diagnosis. And we want to bring those as quickly as possible, but as accurately and safely as possible to our cancer patients. So let's look at a little bit of the history. Perhaps the first clinical trial is actually in the Bible. And King Nebuchadnezzar ordered his people to eat only meat and drink only wine. He thought this would make people healthier. And some people protested and they said they wanted to have vegetables, legumes, and so he allowed it. So this is an open, uncontrolled trial. Everybody knew what they were doing. And when the experiment ended, he said people did well enough that he allowed both to happen. But the first randomized clinical trial, that is where you put different people into different groups, took to 1747 and is ascribed to a physician, Dr. James Lind. And this is in the time where people were going from Europe to the New World by ship. And many of the sailors were coming down with scurvy. So scurvy, we now know, is a lack of vitamin C. All they knew is that these sailors would soon get swollen, bleeding gums. Teeth would fall out. They develop spots on their skin and on their shins, and their skin would bruise easily, and they could no longer work at what they're supposed to do. So what he did is he took a couple of people, and he gave a couple of them a quart of cider, apple cider a day. Two others took elixir vitriol, which is a mixture of different vitamins and minerals and stuff, or we would call elements that they had. He gave 2 spoonfuls of vinegar 3 times a day to other ones. And some people didn't do very well. He gave them seawater. That would not taste particularly good. And 2 others each had oranges and lemon. And it took out that the people who were taking the oranges and lemons did spectacularly well. They very quickly turned around, they healed, and were able to be worked. The day this started was May 20th of 1747, and it became Clinical Trials Day. It's held every year in honor of this, to commemorate the day Dr. Lind began the trial. And of course, he now opened the can of worms that we know as insurance coverage, right? Because he could not recommend the use of oranges and lemons because they were too expensive. And it took almost 50 years before the British Navy would actually allow it. And then they gave lime juice because it was cheaper. So some people had it, but they didn't really roll it out for a long time because of the cost of fruit. So let's talk about the different phases of a clinical trial. We talked a little bit about what a randomized trial is, people getting different things in different groups. And it all starts out with preclinical development. You try to come up with a drug that has some rational reason to be used. And there's many different reasons for that. We won't go into that, but you test them in cell cultures and petri dishes and eventually animal models of disease. Then they enter phase 0, which is the first human clinical trial. So it would be untested. There are ways of figuring out from the animal models what we think the right dose would be, but they give a few small doses of the drug to make certain nothing untoward happens. This is not a therapeutic trial in any way. It's just to make certain that we understand that the way we think the drugs will behave, that's how they behave. And then we go into phase 1. And phase 1 are really studies to determine the maximum tolerated dose, the MTD. And it's essentially a dose versus side effect trial. So we give a certain amount of the drug and you see what side effects you have, and you look to see if it's efficacious, if this is working. Give a little bit more, and you keep doing this in a certain pattern to see if the combination is safe, what side effects, how much we can give. And then if we get a dose that we can give, it goes on to phase 2. You learn more about the safety of it. You test whether a new treatment works for a specific cancer. And then in phase 3 studies, these are the ones that usually go on to trying to get approval, in our case for the FDA, the Federal Drug Administration, a randomized studies to test a treatment. And it worked well earlier. And the idea is that you use either the standard of care, whatever we have. A might be the standard of care, and we're comparing A to B, the new drug, or it could be the new drug in addition to the standard of care. So we want to know if the treatment is better or perhaps is equally efficacious, works equally well, but has fewer side effects. Ideally, we would like both, but that doesn't always happen. So let's talk about some of the other types of ways we figure things out. I'm going to use lung cancer at the beginning as just an idea of a way of like explaining different trials. So the first thing with lung cancer is there were 4 areas of evidence, lines of evidence that suggest that it caused lung cancer. There were population studies. So these are trials, but they're really looking at populations in retrospect. And they looked and they saw that as cigarettes began to be rolled out by machines and became incredibly cheap and easy to use, the lung cancer was rising in that regard. Of course, that's a correlation, not proof. Then multiple studies correlated smoking with lung cancer. And in 1954, the American Cancer Society had a very large cohort study. So what do you do in that? You have some people who you know are smoking, and you take people who are identical to them but don't smoke, and you try to say, well, maybe they have the same exposures because they are both coal miners, or they live in the same area, or they're the same sex. You match them. And when they did that kind of thing, and there's statistical ways of proving that you've done this well, they actually could establish what they said was beyond a reasonable doubt that actually cigarettes caused lung cancer. And they also did animal experimentation. And though I joked to the Gary Larson comic about the real reason dinosaurs became extinct, what they did is they actually gave some of the smoke and inhalation products, or they would take the tars from the cigarette, put them on animals, and saw that it caused cancer. They also looked at how cigarette smoke affected the lungs. It destroys or reduces the ability to clear pathogens from your lungs. And they also found cancer-causing chemicals in cigarette smoke. And so by then, they knew certain things were carcinogens. They caused cancer, and they found all sorts of things in them: arsenic, chromium, nickel, what we call a zoo of what they call polycyclic aromatic hydrocarbons. Many of the things were generated, some by the pesticides that were put on them, but some of it by the burning of the cigarettes at high temperature. So all of these things suggested that lung cancer actually was caused by cigarette smoking. So the first thing that happened was surgery, and then we knew that we could remove it, and most people still died. And there were studies done, randomized studies, looking at the addition of radiation, because some people would recur locally, just in the area where the cancer was. And could we improve that with them? And so in lung cancer, this proved true, that surgery and radiation can often help people who had local lung cancers that were being removed to live longer. And one of the other kind of avenues of evidence of this kind of surgery and radiation came from breast cancer. And a lot of that was done at the NSABP, at AHN, or at AGH at the time, right, with breast cancer, where they used to do very large resections, removals of the breast and the underlying tissue and everything for breast cancer. And then studies were done that showed that you could do a much smaller operation that wasn't as disfiguring, wasn't as morbidity causing, meaning causing side effects, and use radiation and get an equipoise, getting an equal benefit to it. So surgery and radiation were complementary. And then in the 1980s, people started looking at chemotherapy. So if you did surgery and radiation, could you give chemotherapy? And these were single drugs for the most part. And there weren't many that were available at that time. Starting in the 1990s into the early 2000s, so only a couple decades ago, this was the best we had, and there were different chemotherapy combinations. So as an example of this, in 2002, this was a sentinel paper in lung cancer, and it was looking at 4 different drug combinations, and the actual drug combinations don't really matter. They all had different side effects, and you can pick it. But each of these is a survival curve for one of these drug combinations, and you can see they were equal. So it's sort of like pick your poison, almost. But it's kind of sobering, right? Because this is months of survival. So only 20% of the people are alive in about a year. And by 2 years, very few people are alive with just chemotherapy. These are advanced, non-resectable, not surgical candidates. But it will begin to show you how much this has changed. So what has happened with new clinical trials and new drugs? We have learned about targeted therapies. That is a way of figuring out particular changes in the DNA of a tumor that we can target specifically, almost like a lock and a key. And then we've added that to chemotherapy. And then they've come up with new ones. Once you know a little bit about how it works, you can make newer derivatives of it that work better. And we'll also talk about what everybody wants to talk about, which is immunotherapy. And immunotherapy has really only been around for the last decade. So we call these driver mutations that can lead to targeted therapy. So just so that everybody's on the same level here, got to talk a little bit about DNA. And if you know what DNA is, you can turn this off for a second. But DNA is the fundamental biological unit of data storage. So basically, it comprises sequences of 4 different, what we call bases: adenine, thymine, cytosine, and guanine. And they, in different combinations, they can actually code to make proteins. And those go on to be the basic unit of heredity that's passed from parent to child. So a gene gives rise to an intermediate called RNA, the RNA to proteins, and that does all the work or so. So we look for mutations. So mutations are DNA changes that can activate a gene. Activate means turn it on when it shouldn't be, that would cause growth, or be deactivated, such as when you have sickle cell disease or other gene diseases where if you have loss of function, hemophilia is another one. There's lots of different ones that can lead to a risk of a disease happening or lead to the disease itself. So certain changes turn things on, certain things shut things off or remove the brake of it. And if you look at this, cancer is a combination of these gene changes where we have genes that turn things on and make them grow, and the loss of the braking. So I often like to tell patients, it's as if I got you in your car, put a pole and put your accelerator down all the way to the bottom. As long as you could, and put the car in drive, as long as you could step on the brake, it may not be fun, but you'd be able to slow it down. If I cut your brake line, you no longer can control anything. You've got uncontrolled speed or uncontrolled growth. And so driver mutations, that term's used to describe changes in the DNA, the sequence of the genes that allows cells to become cancer cells, to grow and spread throughout the body. So let's look at non-small cell lung cancer. Lung cancer is divided into two non-small cell lung cancers, and some other smaller ways of filleting it. But we're going to look at just one called non-small cell lung cancer. And what on the left shows there's a whole number of genes. I'm not going to tell you what each one of these genes do, though they have names here at the bottom or so. But what happened is that we realized there was a number of people who never really smoked, and especially people from Southeast Asia who were getting lung cancer, and it didn't make a lot of sense. And it turned out, as we were able to sequence DNA, remember the first genome was not sequenced until about 20 years ago, and it was quite expensive and difficult to do it. Now it can happen in a matter of days, actually, to figure out what the changes in the DNA are. And they found that there was something called epidermal growth factor receptor. And it was known that there was a drug called gemfitinib, which actually could block that. And it was shown in a randomized study. People got regular chemotherapy, or they got gemfitinib, and it showed that if you had an EGFR mutated cancer, right, so your lung cancer had that, you did much better with the small molecule. These are just oral pills that you can take. And there's another gene called ALK, and crizotinib was first found to be effective in that. And it was then found to be in a randomized study. So here's an earlier phase 1, phase 2 study where they just looked in ALK altered tumors. And then they were able to do randomized studies that showed it was better than chemotherapy. Then they also found out that there was another gene called ROS, where this gene, small molecule called crizotinib, could also help. And then we got, as we said, newer EGFR-directed therapies. So we're just going to look at this EGFR thing. The next generation are much better. What would happen is that you would become resistant to these. The genes could actually change. It's like having Darwinian evolution in real time. If your tumor, which is growing very rapidly enough in the background, had just a change by accident that made it resistant to gemfitinib or alatinib or so, then what would happen is that the tumor would start growing again, and the new one came out, osimertinib, and it was superior to chemo with failed previous EGFR drugs. And so I'm not going to go through all of them, but all of these other genes now have targets. NTRK has a drug, NRG, RET, MAF, BRAF, HER2, and they're all effective. So you can ask, here's what's called a meta-analysis, looking at lots of different trials and stuff, and looking at events that can happen. And so people ask that if you had stage 4 EGFR mutated non-small cell lung cancer, right? Did you do better with chemo, immune therapy, or because immune therapies were around at this time that this was done, or this TKI, tyrosine kinase inhibitor, which means the targeted drug. And in red, you can see that the oral pill was greatly superior, and that even out, right? This is, looking at 20 months, 30 months, sorry about that, into 3 years, 4 years, and this continues to plateau. Some of these people were, what's going on here? It really went ahead, I apologize. There we go. A number of these, I've got to be careful with my mouse here. A number of these people actually can have very long-term survival with this, which never happens with chemotherapy. So what are the lessons learned here? That clinical trials leading to targeted therapies in one disease is going to turn out can be expanded to include multiple similar molecular profiles. So we sometimes call these basket studies, which we'll describe a little bit later. That is, you might give an EGFR drug and you give it to people who have EGFR mutated lung cancer or colon cancer, or an ALK with lung or pancreas or colon. So all of these things can be fun. And it turns out that the same drugs that might be effective in one will be effective in a whole different cancer. So what we're learning how to do is getting away from this kind of like just hit it with chemotherapy, which has all side effects. Not that these drugs don't have side effects, don't get me wrong, but they're usually better tolerated and they target the actual problem, and they can be used in different cancers. And I'll show you some examples. This is a now about almost 8 years old, a young gentleman who came to me with fatigue, diarrhea, and jaundice, and had a pancreas cancer. And we were just beginning to do gene sequencing. And I got a call from this company called Foundations Medicine on a Saturday night at 11 o'clock, and they were ecstatic. And they had found a change where this ALK gene, which we talked about before, was paired with a whole nother gene. So they were fused together, and that causes it to be active. And so if you look here on the left, this should not be there. That's a lymph node. And there's some more lymph nodes in here. And this is a gentleman who already had his pancreas out. So I can't show you the primary pancreas mass, but he recurred. And we gave him those same drugs that you use in lung cancer that have been proven to be helpful with this. And they shrank away. And he ended up living another 7 years or so through all of this. And so it was a rather amazing kind of response. We're going to go to biliary cancers. These are bile duct cancers. So this is a schematic of the liver, and these are the bile ducts that kind of take all the bile, kind of the waste products and other things out of the liver, and they can get a cancer. And it turns out to be about a third of these actually have mutations, changes, driver mutations, or these translocations where you activate a gene, turn it on because you pair it with something else. And these have all sorts of interesting names, IDH1, KRAS, FGFR fusions, and it turns out that you can actually use the same targeted therapies that we've learned that work in breast cancers or colon cancers or lung cancers, in bile duct cancers as well. And so here's a patient of mine who had gotten all treatments that we had for bile duct cancer. If you're not used to it, this is kind of a normal liver, and this is a big metastasis. This is a cancer in there. It actually, he had another one somewhere else. You can see one out here also. And he went on this drug, pemigatinib, which is approved for these fusion proteins. Happening all the time to me, sorry. And you can see that he had actually a response. All the lesions got better. And he actually, even though he's followed now locally in the Altoona area, he is still responding to this treatment and still alive today, where he would not be alive except for this therapy. So we've talked about kind of targeted therapies, driver mutations. And now everybody wants to know about immunological, immune therapy. If you turn on the TV, you can't hear about how amazing immune therapies are. And for subsets of people, there can be rather amazing, but not for everybody, unfortunately. So just to make certain again that we're all on the same level, talk a little bit about the immune system. So we've got a little cartoon. You have a normal cell here, and you can get gene damage. Remember, we talked about genes, the DNA, and it can be due to smoking, can be due to UV light, melanomas. That's how you get a cancer in your skin. It's the ultraviolet light. You can have inherited or other DNA problems in there. And what happens is when you make these new rearrangement, these new changes in the DNA, sometimes what happens is you get lots of different changes. And the way your immune system works is it's constantly surveilling for things that are not you. And sometimes by random, you damage the DNA and you create, remember, DNA becomes this thing called RNA proteins. You create a protein that actually your body hasn't seen before. And so it can react to it. And so that's called a new antigen. Antigen means something that the immune system can see, neo. And it's processed by what's called an antigen presenting cell. So this is a T cell in your body or so, and it latches onto this and it says, okay, come over here and attack it. Now, there's these things called immune checkpoints, and that you don't want your immune system to go so crazy if it's doing this not for necessarily cancer, or even if it is for cancer, but because you've got the flu and your immune system is attacking that virus or the cold or whatever kind of problem that you have. At some point, your body has to say enough's enough. You're going to start damaging your normal organs. And so there's the immune checkpoints. It makes sense. And they have interesting names. We're going to talk about a couple of them. PD1, programmed death ligand 1, and then programmed PD PD-1 here, which is the thing that can actually bind it. So these two things can bind, and there's many others, but another one is CTLA-4. So these CTLA-4 was the first one was to be found. PD-1 was the second one to really be described in great detail or have a targeted therapy for. And just a few years ago, these won the two discoverers of this the Nobel Prize. So we've been really with these immune systems and immune targets in melanoma for about 10 years, which is where the CTLA-4 and PD-1 and other cancers only for about 5 or 6 years. So now, interestingly enough, the immune system should be able to see many of these cancer cells because some of them will be different. And what happens is cancer cells can take over these checkpoints, so the immune system no longer targets it. And it's very interesting, if you take out a cancer that looks like it, you can find the immune system, these white cells surrounding it, almost like trying to get in there and kill it, and almost like those Roman gladiators that form a little shield around their center or so with their big shields or so, are these little cells trying to block it, the cancer cells or so. And so what these drugs that have come out are inhibitors of this interaction, so the immune system can once again target the cancer. So, as I alluded to, this was first being used in melanoma, and the historical 5-year survival for people with stage 4, meaning it had spread outside of your local skin area, wherever that might be, was as low as 3%. Some people said 10%, but it's incredibly low. And what very interesting, this is a PD-1 inhibitor called Keytruda or pembrolizumab. And they looked at it compared to another one called ipilimumab. And this was not a study that was designed to prove one was superior, even if the pembrolizumab or Keytruda arm is above that. That's not statistically significant. But just to show you, compared to like 3% survival, you've got 30% or 40%. You can combine these two and get a little bit more, really, at 5 years. So we've taken a thing that was essentially a death sentence, and about up to 30% of these people are cured. And so eventually some people will relapse, and there's a lot of work going on, different new immune drugs to try to understand how we can break that tolerance, once again, that the cancer is caused to the immune system. But this was just rather amazing. When I was in my fellowship, melanoma was one of those diseases that no one really wanted to tackle, right? Because people died in months, and now people are living an amazing amount of time. As a matter of fact, one of the first people on the first trial for Memorial Sloan Kettering on this drug ipilimumab was given months to live, and she's still alive today with no evidence of disease. So these are called checkpoints, as we said, because they're supposed to be points in the immune system to check it from harming anything else. And so they started looking at new diseases. These were out there, right? So these were programmed death 1 and MDX1106, and these are anti-PD-1 antibodies. They were given different names then, but they looked at colorectal cancer. So in one study, they had 39 patients who had kidney cancers, bladder cancers, a couple melanomas, and 14 colorectal cancers. And in another one, 296, of which 19 had colorectal cancer. So that's 33 patients. And one of those 33 patients, none of them responded except one did, had a complete response. All their tumor went away. And that was just rather amazing. And everybody was caught scratching their head and figuring out why was this. And then somebody noticed this patient had something called Lynch syndrome. So they had a deficiency, they had a gene mutation, a change in something called mismatch repair. So I'm going to talk about Lynch syndrome so we can understand this a little bit better. It causes colorectal cancers as well as endometrial, which are uterine cancers. It can cause cancers of the ovaries, stomach, small bowel, pancreas. And it's due to DNA repair deficiency. So DNA constantly is harmed and is constantly repaired actually by a set of enzymes. And so this deficiency can lead to many, many, many changes in the DNA. And so what happens is there's a lot of these new antigens being seen because there's a lot of random reassortment of DNA. And we term that deficient mismatch repair. And so the first thing that happened is people looked at this drug, Keynote 177, which was pembrolizumab, at that time called MK4075, versus chemotherapy. And they asked if you had this deficient mismatch repair. So these people could have had Lynch, but you could also have had tumors where they just lost the ability to do that mismatch repair outside of the hereditary syndrome. And we can tell the difference, but at this level, they're essentially the same thing. And you can see if you gave people chemotherapy, there were very few, about 30% were alive in a couple of years. And a number of these people here, if they got the immune therapy alone, did significantly better. And you look at this and you just go, oh, wow. Because eventually, if you take this out to 4 years or so, if this had gone, this continues going off the scale here, and they're all dead, but these people are still alive. So this was a basket trial that was done. So we alluded to it before, and they asked if all of these different tumors you had in the GI tract, cholangiocarcinomas or biliary tumors, prostate, small intestine, thyroid, had mismatch repair, you could screen for them. And if we gave all of these people, any of them, pembrolizumab, what happened? This is called a waterfall plot. So if you take a look at the tumor size, a few people didn't respond, but most, had kind of stable disease, and the vast majority of people responded, and a significant number, the tumor went away. And this was just eye-opening, because this led in 2017 to the first tumor agnostic approval. Prior to this point, even though we talked about targeted therapies before that, it was approved for EGFR mutated tumors in lung cancer. It was approved for ALK mutated tumors in lung cancer or pancreas cancer. The FDA approved this drug, pembrolizumab, for any tumor anywhere with mismatch repair. And so it really opened up a lot of new trial designs and things that we could do that we couldn't before. And so I'm going to give you another example. This is a gentleman who I had who had cholangiocarcinoma, biliary cancer. We had treated him with chemotherapy 1, chemotherapy 2, chemotherapy 3, and he failed all these things, went on a phase 1 trial of new drugs, failed that or so. And I referred him for biopsy back in 2013. This was just becoming available, this idea of doing genetic sequencing or so. I put him on hospice and let's see what happens. We'll do this biopsy. And I got this result back that he actually had mismatch repair. Now, he had that type of mismatch repair that was not inherited. Nobody in his family had it. But I was able to get him on a trial. It was then opened at Johns Hopkins. We brought it to where I was then. We got him on this trial, opened it very quickly. And this is his tumor marker that kind of found the disease. And you can see it just, this is what it was doing, rising on all chemotherapy. And then it just plummeted and it stayed normal. And his tumor went away. And he has been on this drug since 2014, completely off hospice, living a normal life, this long-term, long-distance truck driving, and just doing amazingly well. So let's go back. We're going to cycle back. We talked about special cases. Now let's try to talk about broadening it, non-small cell lung cancer. So there's a test. You say, sometimes can we figure out who might respond better than somebody else? And remember, we talked about this programmed death ligand, and you could assay it. An assay is just a matter of taking a look at the cell and trying to figure out how much of this, what percentage of the cells have this on the surface. And it turns out that when they gave checkpoint inhibitors to patients with non-small cell lung cancer, if the percentage of the cancers that had cells, so the cells that had PD-L1 was greater than 50%, these patients did incredibly well. And if they had a lower amount, they didn't do as well. So we can derive, the lesson from here is by doing a trial and looking at blood and other markers, we can begin to derive ideas of who might benefit from one drug or some combination over another. So let's take this to a new trial to the next step. And this was looking at pembrolizumab, that same PD-1 drug, plus chemotherapy and metastatic. So this is non-small cell lung cancer that is spread. And if people had greater than 50% of their cells, remember that was the number that was derived earlier, you could see that they did incredibly well with pembrolizumab plus the chemotherapy. And they didn't do as well with placebo plus the chemotherapy. But even if they had 1 to 49%, they still got some benefit. And even when they had very low, there was some benefit to it as well. But this has led to, if you consider, remember, there were responses in those people who had greater than 50%. Some people who can't get chemotherapy, but you do this test and you look at their lung cancer, right? And you say, they have greater than 50% PD-L1, more than 50% of their cells have this on it. You could offer them an immune therapy with far fewer side effects. And you can be sicker, you could be older, and you can probably tolerate this. And so now you've created a paradigm where we previously talked about in non-small cell lung cancer, that before you want to give chemotherapy, you're interested in finding out whether they have these driver mutations. So we have to get the DNA out of get a tumor sample, send it for sequencing to figure out if there's any driver mutations. And we want to look at this PD-1 score, and then they can get to the oncologist, then they can get to the surgeon if they need to decide what the best treatment is. So here's another lesson. Can we move treatments to earlier stages of cancer? So remember, we talked about mismatch repair, patients with Lynch syndrome or a Lynch-like syndrome with colorectal cancer. So this was done in rectal cancer alone, and you might have heard about this on the news. And what usually people with rectal cancer have to go through chemotherapy, radiation, and many of them end up with a pretty morbid surgery. They can remove part of the rectum, sometimes all of the rectum, and you have a permanent bag, and no one wants that if they can avoid it. And so the thought was, since they respond really well to these immune drugs, can we give them a PD-1 drug or so and see what happens? So the idea was to give this to them for about 3 months, image them. If they still had disease there, you could go on to chemoradiation. If they still had residual disease, you'd go on to surgery. But if you had a complete response, they would just follow you. And so this was kind of very intriguing. By moving it earlier, turns out this paper is actually from 2022, talked about the first 12, but at a recent meeting, they're now up to 20. And so all the patients who had this subtype of rectal cancer, who got this PD-1 drug, none of them needed surgery, which is kind of mind-blowing because we don't think about that. So there's something about giving these drugs earlier that might not breed the resistance than later. And there's some theoretical reasons to think that if you let the disease get bigger and metastasize, they may not respond as well to these drugs. But this is kind of a proof of principle. And this has led to that if you have a rectal cancer, before we consider treatment in our multidisciplinary rectal clinic, you're going to go on to get this testing to see if you have this subtype. And yes, we have found a couple people who have. Yes, we've treated them like this. And yes, they have not needed surgery, which is really amazing. So this is some recent data looking at giving neoadjuvant, nivolumab is another immune therapy plus chemotherapy, and looking to see if you gave it prior to surgery in people who have resectable. Resectable means it's removable. Instead of removing it and giving chemotherapy afterwards, as we often do, or immune therapy sometimes afterwards, as we might do in certain situations, how about if they gave it earlier? This doesn't prove that the survival was better, but this showed there's a tendency towards, at least here in the looking of people who got it neoadjuvantly, meaning prior to surgery, did better than those people who got chemotherapy alone. And so once again, this kind of shows the paradigm of trying to do studies where you change when you give a drug, prior to, afterwards, before and afterwards, as we're going to see about this. And this was a trial in melanoma, where people had proven already that if you had a melanoma and it was of a certain type, it was a certain risk of recurrence. If you gave pembrolizumab, this PD-1 inhibitor, for a whole year, people did better than if you didn't give them, if you gave them a different drug or so. So there was a survival benefit. So they asked a question in this trial of if you gave it neoadjuvantly prior, right? And that, or if you gave it adjuvantly, which was better, right? Before or after. And this one only was powered for event-free survival. Overall, it's not mature for overall survival. It may be that people who got it neoadjuvantly go on to relapse at a different rate. We don't know, but isn't it intriguing that if you give it earlier to patients with melanoma, that they appear in the short term to do better? So we've talked about immune therapies, right? And things that we've generated and targeted therapies. Now we kind of want to switch a little bit to biological therapies. So you've probably heard about these. These are also within the realm of immune, but what we're doing is actually taking cells from a patient, altering them, what we say ex vivo, outside in the tube, in a machine, and going ahead and giving them back to the patients to fight the cancers. So the first one that I'm going to lose, this one on the left, are called TIL cells. So if you take a look at a tumor, you exercise part of the tumor, lymphocytes are white cells, and we know that there's white cells inside there that appear to infiltrate the tumor. So we call them tumor infiltrating lymphocytes. You can isolate those. You can expand them on a machine and in dishes and bags and stuff. You can test whether against some of the tissue, will they kill the tissue? And then you can give them back to the patient. So you have to give some chemotherapy so they'll be accepted. But the idea is you take this out of the patient, you isolate the good portion of it, give it back to the patient. And this has proven incredibly helpful in certain patients, as we'll begin to show you. But the other way of doing this is taking T cells, they're called CAR T cells, or chimeric antigen receptors. So T cells have on them, we talked a little about when we said antigen presenting cells, they have on their receptors, they have little things to recognize the cancer or so. And so you, in this way, you take the T cells from a patient, actually from their peripheral blood, and you actually modify it. And then what you do is you add it in, you add something into it, which has gene things and a virus which can multiply in there but can't infect the patient. And we'll add something to those T cells so that they're better able to fight the cancer. They're more specific for the cancer. You expand them and then you give them back to the patient. And there have been amazing things done in both of these where CAR T cells, this thing, has truly changed the landscape of many B-cell lymphomas and T-cell lymphomas, where this is now a standard protocol. And something that we're exploring ourselves at West Penn Hospital, which is an AHN where we're doing all the bone marrow transplants and stuff. And there's a number of TIL trials as well that we'll show you. And so this is a phase 1. Okay, remember phase 1, again, safety and tolerability. This is a TIL trial from a company called Nurex, and this is a woman who had advanced gynecological malignancy. And she had a number of tumors from ovarian cancer. She had her tumor taken out, some TIL cells made, and she had an excellent response. And of course, no one knows exactly what the long-term response is for this, but that's not necessarily the idea of the phase 1. Remember, this is safety, tolerability, and to tell us should we continue on this vein or not. Here is a recent publication, right, just from a month or two ago, was headlined at the SITC, which is Society for Immune Therapy and Immune Cellular Therapies. It was tumor infiltrating lymphocytes. So remember, this is you take the tumor out. These are patients with advanced melanoma who had failed pembrolizumab or nivolumab, this PD-1 drug, right? And the standard was to give a different one, the CTLA one, the one I told you was first found, ipilimumab. And they asked, if we take these out, these melanoma cells, create TILs, figure out which ones are fighting the tumor, give them back, do you do better or worse? And it turns out they do significantly better than those patients who got the standard of care. And once again, we call this kind of a tail on the curve, but these people all eventually relapse. About 25% of these people are still alive. So people who failed therapy at the beginning and relapsed, we now have an alternative at these TILs for them. And this is at the FDA being looked at to be approved as a therapy. However, we've got a number of active clinical trials. There's a number of people headed by Dr. Yazin Samuri and Dr. John Lister and David Bartlett, who's looking at viral vectors and viruses that can fight cancers. In this trial, an early phase 1 and phase 2, including expansion phases for melanoma. So this trial is available now at AHN for patients who have failed melanoma. And you can see it's being looked at a lot of different tumors. And we think that this type of thing is really going to be able to change. Not only is it helping people, we said, with B-cell, with lymphomas or leukemias, we're trying to learn how to use this to help people with solid tumors. And as a matter of fact, the first patients with lung cancer are being treated this way, and the first patients with colon cancers are being treated by Dr. Samori and Dr. Lister with this at West Penn Hospital. So this is just to look to the future a little bit. And we talked about these immune checkpoint inhibitors and what the CAR-Ts and the TILs are adoptive cell transfer. But there's also cancer vaccines, which we don't really have time to get into, but these are being tested, and oncolytic virus therapies or so as well. And this is something that Dr. Bartlett has spearheaded here at AHN, with the idea that you can create a virus that can actually lyse or kill the cancer and direct it towards it. And yes, you can use these in different combinations as well. And so all of these, you're going to be hearing about more of these in the year to come. And each of these have to go through a clinical trial. Phase 1 to prove that they're safe, what the tolerability, and then if we can prove they're safe and effective, we can begin to combine them in novel ways. So what have we learned from our clinical trials? So they've uncovered novel mechanisms of treatment. There's the novel classical chemotherapy or surgery and radiation combinations, as we talked about in lung cancer and breast cancer, but it's true for all sorts of cancers that we can combine these things. Targeted therapies, those that have driver mutations, immune therapies for both specific and all more normal, widely available for more people with like lung cancer and other kind of cancers. Combinations of all the above, people doing chemotherapy, radiation, targeted therapies, immune systems. Again, they go through trials. And cellular therapies now is going to be the next generation of these. And we're beginning to unravel mechanisms of disease and treat underlying cancer pathology, not just the cancer. It's not just drug A and B for this tumor. It's actually targeted therapies across cancer types, immune therapies across cancer types. As we're learning that immune therapies or targeted therapies or combinations of these chemotherapies and these things can be effective in one area, can we move them to earlier aspects or later aspects of treatment? So we talked about lung cancer and melanoma and colorectal, all where this paradigm is being explored and it's being further explored as well. And unfortunately, I don't really have time, but the best thing for cancer, to treat cancer, never have it happen, right? And so my plug for screening, AHN has a free screening clinic that goes on once a month all across the network. And but you can get it through your insurance too. And there are screening tests for lung cancer for people who smoke. And it found that if you use these screening techniques, you actually have a better survival. You can find it earlier. Breast cancer with mammograms, colorectal cancers with colonoscopies, high-risk pancreatic cancers, and more are coming online. And so it's really important when you see your PCPs, you wear a seatbelt, right, when you're in the car, because it improves your safety if you get in an accident. Well, this can keep you from dying from a cancer. All of these were done in trials, large trials of colonoscopy versus nothing, and people who got the colonoscopies lived better. Colonoscopies versus stool DNA testing. And, you could say whether there's differences in them. Breast cancer, mammograms across different age groups. We can argue exactly whom to do them in and who not to do them in. PSA testing in prostate cancer is another one. We can argue exactly where it should be, but, they're there and you should take advantage of them. All of them were found by clinical trials. So as we head towards the end, what I want to do is just talk about, different types of trials. To remind you, we had basket trials. We talked about targeted interventions across multiple diseases. So this might be a EGFR inhibitor in lung cancer, pancreas cancer, bile duct cancer. Umbrella trials where you might just look at people who've got breast cancer, give an intervention 1, an intervention 2, an intervention 3, and see how people respond. So they're randomized to different ones, perhaps based upon their types of mutations even. And what we call a platform trial. And these are becoming much more evident, especially amongst pharmaceutical companies and cooperative groups. Cooperative groups are groups of oncologists and radiation oncologists and nurses and clinical research coordinators who all get together and actually combine to try to create new novel trials and bring them to the public. And so a platform trial is you can have standard of care, and you might have intervention 1 and intervention 2, and you may introduce a new arm, something new came up and you want to add that. This arm proved not to be as good as the standard of care, so it's dropped, and you can keep doing this over until you get a final analysis. So what you do is you have what's called interim analysis. You say, we want a response rate of a certain amount because we know that that response rate will correlate with survival. And so you can begin to try different things, reiterate them, change them, add arms, these are called arms of a trial, and be able to change them. You do these within cooperative networks, of which we're part of them. There's the Allegheny Health Network and all these other major oncological cancer centers as well. So one of the other things, kind of like, the future of what we want to look at as well, is methods to improve the efficiency of the drug approval process. So we have these N of 1 clinical trials, such as my young gentleman who needed and had an ALK mutation in pancreas cancer, or my gentleman who got the MSI, the mismatch repair, it's also called MSI high, cholangiocarcinoma, and we were able to get it for him. Right, that was kind of an N of 1. We made it into a clinical trial, so we report it back to the FDA. But you can also get off-label treatments for this. So if you had a cancer, there's a small soft tissue, a sarcoma that had one of these, you could try to apply, and most insurance companies would say yes. Expanded access programs, as we are involved in with TIL trials for melanoma, the company's saying we want to make this available before the FDA approves it so we can get it to people quicker. Decentralized trials, as I had on the previous slide as well. And I would be fair if I didn't talk to you about one of the next generations of things that we're trying to do. And this is close to our heart at Allegheny Health Network at the Cancer Institute, and is the Moonshot Protocol, which is the brainchild of David Bartlett, who's the Cancer Center director. And what this is people come in and they're, out of the generosity of heart, saying we could look at their baseline tumor, look at their blood, look at proteins, cancer markers, DNA in the blood and the tissue. And every time they come in, like about once a month, they allow us to draw more blood. We can collect all this data, correlate it with the medical record, and try to figure out, why did person A respond? Why didn't they respond? Why did person A get bad diarrhea? Why did person B's hair not fall out? And so the other thing that we can do is, if we can figure out enough of these, we might be able to create a panel that you can begin doing blood-based screening. That would be the, je ne sais quoi, of like, of cancer, right? Could we do a blood test that would detect cancer or so? And so this is an ongoing thing. We're trying to get up to about 10,000 patients, and there's a huge infrastructure to do this. So with that, I'd like to end and open it to questions. And I would not be fair if I didn't acknowledge all the patients and their families who've participated in these trials, going all the way back to Dr. Lind on that boat coming across to the New World and scurvy, to everybody today. I mean, people are volunteering for these. Many people gain benefit, absolutely, but much of it is done out of the generosity of them and their family's heart to help somebody else. So I can't thank them enough. And I thank you for your attention.
Thank you, Dr. Bahary, for that very informative and interesting presentation. We do have time for 1 or 2 questions. I have one in the chat that says, is there as much focus on gynecologic cancers as there is on lung, colorectal, etc.? And it's just not discussed here, or is there an overall lack of focus on gynae cancers?
No, it's a wonderful question, and I'll apologize to Dr. Krivak, who takes care, who's the head of the GYN oncology, is one of the people whose pictures I put up there on the board there of people doing it. And there are a lot of good GYN. I just it's somewhat a lot of these same things are going on. Targeted therapies in people who have BRCA mutations with ovarian cancer. Also, they can get pancreas cancer and others. And so there's drugs now targeting that. There's people looking at TILs and immune therapies. And there's been wonderful things with endometrial cancers and immune therapies. It was just so much going on in this talk. I just left certain things out. I also didn't talk about brain tumors and immune therapies and sarcoma. So I apologize if, but it was not an intended leave out. It was just time-wise.
Thank you for the question. How about we have time for one more? Are there any new exciting clinical trial designs coming soon?
Yeah, as I said with these platform trials, a lot of what we're trying to do are called window of opportunity studies. That is, if you give immune therapies like neoadjuvantly prior to surgery for cancers that we know don't necessarily respond, can we then get the whole cancer out at surgery? So you give it for some period of time, it's a window of opportunity, and try to understand what the changes were in that tumor and why they did not respond or why they might respond, so you can go back and tweak it. So can we learn who might respond? So more and more clinical trials are looking at prior to surgery for little periods of time with novel agents in order to understand, because you can get the entire tumor out and do many things with it. So they're called, window of opportunity studies, because you have this little window to study the drugs before going to surgery.
Great. And we have one more quick last minute question. Do you have any current clinical trials specific for neuroendocrine cancer?
There is a trial for neuroendocrine cancers for people who failed first-line therapy with a drug called Afinitor. It's looking at a new drug called cabozantinib, so that's available for certain people. And we're looking at bringing on and growing our nuclear medicine. And there's a couple of new trials for neuroendocrine tumors working their way through the regulatory mechanisms and stuff so that we can open them up in the future for patients with neuroendocrine tumors.
Great. So there's just a comment, great lecture. Thank you, Dr. Bahary. And at this time, we'd like to thank all of you for attending our Ahead of Cancer Lecture Series event. Please continue to join us at our monthly Ahead of Cancer series as we focus on cancer screening, innovation, detection, treatment, and survivorship. As a reminder, tonight's lecture was recorded. If you're interested in re-watching tonight's lecture, please check back on the AHN YouTube channel at www.youtube.com/Allegheny Health Network for the full recording. This video, as well as other past lectures, will be posted under the Ahead of Cancer playlist. If you would like to schedule an appointment or have questions regarding a potential cancer concern, please call our Hope line at 412-578-6473 to schedule an appointment. You may also call our 24-hour Nurse for You line at 412-687-4968. Thank you again for attending.